All right, so for the first part of the video, I went over some key components of a plasmid, and now I'm gonna show you how, basically an overview of the entire lab and how it would all come together and what kinds of results we would expect. So for this video, what I would like you to do is, I want you to get some paper out and pause and play throughout, because I'm going to put questions up on the screen. And so whenever there's a question, I want you to, in addition to taking notes, answer the questions and then like pause and then play when you're ready to move on. Okay. So all right. So this, starting with the big picture. So with the Amgen Lab, what we were going to do is the whole point was kind of putting together all these tools in biotech and like you guys actually doing them. And then, but the big final product was to transform E. coli cells to produce red fluorescent protein, which is abbreviated RFP. So RFP originally came from this like coral. And so they isolated that protein, and that gene, the idea was we were going to insert it into E. coli cells so that instead of E. coli, when they grow, each of these dots, like I mentioned in the last video, these are all colonies. So each of these E. coli cells, if they get transformed, they would produce RFP, which would make them look pink. So this was a picture from actually last year's class. Every little pink dot is one E. coli cell that was transformed and then overnight replicated and multiplied to produce a colony. So starting from the beginning with this lab, there are actually two plasmids that we used, or that we would use. So pause real quick, and I want you to actually sketch each of these plasmids and kind of mark off the components. I'm going to go through each one once you guys have it down. Okay, so pause it, copy. All right, so with these two plasmids, PCAN-R is the name of one plasmid, and then PARA is the name of the other plasmid. So as you can see on these diagrams, they tell you how big the entire plasmid is. So PCAN-R is 5,512 base pairs. So this entire thing is 5,512 base pairs. PARA is 4,872 base pairs. So there are some components on here that some you might recognize from the last video, but some might be a little bit new just because of the way they named it. So let's start with the PCAN-R. So with the PCAN-R, this plasmid is the one that has RFP. So RFP, this is the gene that we're trying to isolate and insert into E. coli cells. So RFP would make the protein that would turn the bacteria cells pink. Next to it is a green section labeled PBAD. So PBAD, this is actually the promoter. So write down, jot this down. This is the promoter sequence. It's just named PBAD. It's a specific type of promoter. So from what we learned about transcription, the promoter is where RNA polymerase would bind. So RNA polymerase would bind here, and then it would then transcribe the gene next to it. So if you notice, the arrows are pointing in the direction the gene is red. So if RNA polymerase binds at the promoter, then it would continue and then transcribe RFP into mRNA and then into the protein. Then you also have this yellow sequence right here. So it says CAN-R. The R stands for resistance. So this is an antibiotic resistance gene. It is resistant to an antibiotic called kanamycin. So this plasmid, if a bacteria cell has this plasmid within it, it would be resistant to kanamycin. And then the last parts of this plasmid is you have BAMH1 and then you have HIND3. So these two lines, these are actually restriction sites. So just as a recap, BAMH1 and HIND3, these are two types of restriction enzymes. On this chart right here, they show you three different restriction enzymes, and they show you the source. So all of these restriction enzymes came from different bacterial cells, and they are named based off of the bacteria that they were discovered in. So EcoR1 came from E. coli. BAM H1, HIN3, they came from these two bacteria cells. And the reason why bacteria have restriction enzymes is because it's kind of like their defense mechanism. So it cuts up foreign DNA. It's kind of like their immune system. So if a bacteria cell got infected by a phage, for example, a bacteriophage, this foreign DNA from the virus, these restriction enzymes could, in theory, attach to the viral DNA and cut it into pieces so that the bacteria isn't affected. So that's how they evolved originally. Each restriction enzyme is specific to a recognition site. So recognition site, we also call it restriction site or cleavage site. They cut at a very specific sequence. And I mentioned this before, but it's a palindrome. So the way you read it in one direction would be the same way you read it in the opposite on the bottom strand. And that's because in DNA in a cell, you don't have like a top strand or bottom strand. It's just there. So they read this specific sequence and then they'll cut. So ECOR1 will cut between the G and the A, and then it'll also cut between this G and A. So you'll have two overhanging pieces. We call those sticky ends. All right, so in this plasmid, you have those two restriction sites. On PARA, the other plasmid, you have some other components that we talked about in the last video. So starting up here, we have the ORI region. This is the origin of replication. You have those two same restriction sites, so BAM H1 and HIN3. And then you also have AMP-R. So remember, the R stands for resistance. So this is another antibiotic resistance gene. In this case, this plasmid would give you resistance to an antibiotic called ampicillin. The last part of this plasmid is an ARAC gene, and I'm going to come back to this later on, so we'll go back to this. Okay. All right, so the first question that I want you to answer would be, number one, which plasmid will allow a cell to grow on a plate that has kanamycin antibiotic? Okay, so for every question, just pause and then restart once you have your answer. I'll post the answer key later on. Number two, what do the arrows show? So if you look at each of these genes, they have arrows. What do those arrows, what are they trying to show you? Number three, what restriction enzymes are used to cut both plasmids? Number four, if both plasmids in the image above are digested, how many DNA fragments will you have? So if these plasmids were mixed in with restriction enzymes and they were broken down, so it's a restriction digest, how many DNA fragments would you get? Give you a hint. 4. So you would have this piece, 1, 2, and then on here, 3, and then 4. All right, number 5. So since I gave you the answer to the last one, number 5, what size will each DNA fragment be in base pairs? So if you look at this plasmid, it tells you the size of the entire thing is 5,512. And then if you look right here, they tell you that this fragment, this piece, is going to be 807 base pairs. So given that information, you can figure out what would the length of this fragment be. And then same on the PARA, they tell you the total size, and they also tell you the size of this smaller fragment. All right, so do you have your answers for that last question? Make sure you tried to figure it out and do the math. Okay, because here are the answers. So if you have 4 fragments, you are going to end up with a piece that is 807 base pairs. So this piece right here would match up to this piece on the plasmid. And then you have this larger piece, which would be 4,705 base pairs. And you would just do that by subtracting. So this top part is the PCAN-R digestion fragments. And then this bottom part is the PARA digestion fragments. So you would get 4 pieces, 4 fragments, and then you can predict the length of each fragment. If you look at this diagram on the sides, you can actually see the overhanging sticky ends because this was cut by hin 3, so it leaves this sticky end right here. And if you look at this piece, it would complementary base pair with this end because this is where it got cut. And then on the other side, you have the sequence that's cut by BAM H1, and this would match up to this piece right here. So that is just what they're trying to show you with these extra bases on the end, is those are the sticky ends that are produced when you use those restriction enzymes. All right, so the next question is, if we ran a gel electrophoresis, how would these DNA fragments look on the gel? And as a recap, we talked about gel electrophoresis really briefly when we were doing DNA profiling. The idea of gel electrophoresis is it separates DNA by size. So this is what a gel would look like. On the very top, these rectangles, we call these wells. They're basically indentations into the gel, and that's where you would pipette in your samples. And then this very first column is the marker. We also, when we talked about capillary electrophoresis, we called it the standard, the standard, the marker, or sometimes we call it a ladder as well. And the purpose of this is they make a known DNA fragment size, and they mix them all together so that you can use this marker column to use it as a comparison. So we know all the DNA that's found in this band is 1,200 base pairs. We know all the DNA found in this band is 1,000 base pairs. So based off of this marker, we can compare these other fragments and predict the size. So this fragment, we would predict, since it is the same position as this one, we would predict that all the DNA right here is 1,000 base pairs. We would predict all the DNA in this fragment would be 800 base pairs, and then etc. So the purpose of the marker is something that we can use as comparison. The way gel electrophoresis works, the reason why these DNA fragments separate by size, and if you look, these are bigger. The ones that are bigger are closer to the top. The ones that are smaller are closer to the bottom. The reason for this is because now this diagram is showing the previous picture, but just kind of flipped in a cross section. So the wells right here are here in this diagram. The well, you insert the DNA, and once you turn the electrical current on, the DNA, because it's negatively charged, it's gonna try to travel to the positively charged end. So when DNA is added to the well, the fragments that are bigger don't move as quickly as the fragments that are smaller, because the gel, agarose gel, it's kind of like hard to get through. So they're trying to draw it in this diagram to show you that it's kind of like a maze. So the fragments have to weave through the gel in order to get through it. So smaller pieces will get further than larger pieces. The analogy I like to use with the 9th graders is if you had, like in this spy movie, and they have those lasers and you're trying to get through it without touching any lasers. So if you have like a laser maze or whatever this is called, and you're trying to get through it, who will get past, or who will get through this maze faster, the little kid or someone big like Shaq? So if you have to get through all these tiny little gaps and make it through without touching any of the string, obviously a little kid's going to get through this quicker than a bigger person like Shaq. So if you look at this diagram, the well's on top, this big piece of DNA, this is kind of like Shaq, and the little piece is like the little kid. So the little kids can move through the gel quicker than a bigger piece. All right, so using all that background, you have these fragments, and they tell you the size right here, and you should have it on your own paper. I want you to draw what it would look like on this diagram. So this is a picture of what a gel would look like. On the top, draw in 5 wells. So these rectangles are the wells. M stands for marker, so this is the DNA marker. once again, you have these known fragment sizes. So number 1 to 10, number 1, this piece would be 10,000 base pairs all the way down to 500 base pairs. So P, can, R digested, these two fragments, based off their size, draw in where they would be on the diagram. So if you look at this first fragment, it's 807 base pairs. If you look, kind of find where it would fall, this fragment's 1,000, this one is 500, so I would predict that 807 would be somewhere around here. And we would just draw it like a line or a band. That would be what it would look like on a gel. So go ahead and sketch in what these fragments would look like on the gel. The first one is for P, can, R. The second column would be for P, AR, A, so the fragments down here. This is an actual gel from last year. So once we ran it, the DNA is stained with a chemical so that when you put it on a UV light, it shines and you could see where the fragments are. If you look at our marker, there are 10 bands in our DNA ladder or the marker. And if you look at this diagram, you actually can see those 10 bands right here. So this was 2 groups. I had 2 groups run their DNA on the same gel. So this is one group, this is the other group. Their bands are gonna look different than what you drew here because they ran different samples, but the marker is the same. All right, moving on. So when you have these 2 plasmids, in the lab, we have these 2 plasmids, but we wanted to actually combine them together into a recombinant plasmid. So in the lab, what we did, or actually not what we did, Amgen did it for us and we got this recombinant plasmid, but they took, let's see if I could write here. So they took this fragment right here and they are inserting it right here. So instead of this fragment, so we would actually get rid of this BAM H1, this section. Ooh, this is messy. So we are basically trying to insert this piece into this little section right here. So if I take this piece and combine it with this plasmid, you know, swapping out for the 377, so I actually put an X right here. Ooh, that's really ugly. All right, but if I was to combine those 2 pieces, number 7 is how many base pairs will the recombinant plasmid be? Let me see if I can erase some of this because it's ugly. All right, let me try to redo that. Well, okay, no, this piece, swap it with, so we're taking this one out and we're gonna put this piece there instead. Okay, so number 7 would be how many base pairs would this new recombinant plasmid be? Make sure you have your answer before you go on. Okay, so in our recombinant plasmid, this is the plasmid when you have those 2 sections combined together. Whoops, this would be what it would look like. So your answer to the last question should have been 5,302 base pairs. Now this new recombinant plasmid, we called it PARA-R-R. So the R stands for recombinant. This is the plasmid that we actually are going to try to now get into the E. coli cells. So going back to our gel, if we were to run a gel using this plasmid, I added a next column on diagram. So this next column, PARAR, which is going to be this plasmid, how would the recombinant plasmid look like on a gel? So if this is your size, draw in where the band would be on your diagram. And then for the final column, what would it look like then if it was digested? So we took this whole plasmid and, you know, broke it down into pieces or digested it. So now we have 2 pieces. We're gonna have this piece and then this smaller piece. In the final column of your diagram, I want you to draw in where those bands would be. So you might have to, well, you can go back and look at your calculations from earlier. Draw in what the bands would look like if this plasmid was digested. Now, what you just drew, these columns right here, the last two, this is actually what we ran in our lab. So that picture that I showed you a couple slides ago, this is actually what we ran. This was the marker. This column is the plasmid that's not digested, so not broken down. And then this column is what you would predict if we broke it down and digested it. So two pieces, one piece, I don't know what it would be, like 4,500 or something like that. And then this piece right here, which would be 807. These are the two fragments here. Now, if you notice, the plasmid, the one that's undigested, it's really high up. This top band, this biggest piece of DNA in our marker, that's supposed to be 10,000 base pairs. But we know this entire plasmid is only 5,302 base pairs. So why is it that it's higher up than this 10,000 fragment? And the reason why is because even though a plasmid is less base pairs, because of the shape, plasmids can come in different configurations. The first most common type of configuration is called supercoiled. So what you do, or what it looks like, is you have the plasmid that kind of twists up on itself, and this would be the most common. It travels quickly for its size, but it's still not super fast because it's kind of a twisted up piece of DNA. All of these markers right here are linear pieces, so it's just one long piece of DNA, whereas plasmids are a circle. So because of its shape, plasmids don't travel as quickly as what you would predict if they were linear fragments. Now the digested ones, these are linear, so that's why they actually are pretty close to what we would predict. So this is the 807 fragment, and this would be the 4,500 fragment. And if you match it up to the marker, that's close to what we would predict. But the plasmid is way higher because of the shape. So you could have it supercoiled. You could have it as a nicked circle, which is a plasmid that's circle, just one circle. And then you can also have a multimer where you have two plasmids kind of hooked up together. So this would be double the size. It's already 10,000 or so base pairs. And because it's not linear and it's an open circle, it would move pretty slowly. So that's why you have the plasmids higher up than what we would predict. All right, so we did not digest it though. We're trying to make the recombinant plasmid. So just to show you the ligation, what this would look like, this red piece is the piece that has RFP. This blue is the rest of the plasmid that we're trying to attach the RFP section to. On this end, you have the BAM H1 sticky end, and if you notice, these bases will complementary base pair with this side. And then on this side, you have the HIND3 sticky end. So this sequence TCGA is going to base pair with AGCT. So when you are ligating these pieces together, you kind of mix them together in the same culture. The bases will complementary base pair, so match up. These green little oval things, those are ligase. So that's an enzyme that will seal and form the bond in the backbone of the DNA. So the phosphodiester bond right here. And then now you have your recombinant plasmid. But we skipped all of this. Basically, we are getting the plasmid already made at this step. We are gonna get this pre-made from Amgen, and then we are now going to try to transform the bacteria with those. So with ligation, once again, here's the animation, and then ligase coming in to form the bond. The next question for you to answer is number 10. Why do you think it is useful to use two different restriction enzymes versus just one type of restriction enzyme? So my hint for number 10 would be, think about what the arrows mean on the plasmid diagrams. Earlier, I had you answer, what do those arrows, the directions mean? Well, why would it be useful that we have two different restriction enzymes versus using the same type of restriction enzyme on both ends? Okay, number 11, moving on. Cells transformed with PARAR will be able to grow in the presence of what antibiotic? So based off this diagram, the plasmid diagram, or the plasmid map, what antibiotic would these bacteria cells be resistant to? And lastly, I skipped over ARAC. So now I'm going to come back to it and tell you what this final gene is for. ARAC is a gene that is part of the arabinose operon. So before we left on this shutdown, we talked about operons and how prokaryotic cells regulate their genes through operons. So arabinose is very similar. The arabinose operon is very similar to the lac operon, the lactose operon. So go back in your yellow packet and look it over if you need to. But the idea was lactose and arabinose, and you look at ose, that tells you it's a sugar. So arabinose is a sugar. These operons, they are inducible, so they're normally off. But if arabinose is present or if lactose is present, they get turned on so that transcription can start. So if arabinose is present, which is a sugar, the RNA polymerase can bind to the promoter, which on our diagram is called PBAD, and then transcription is turned on. So this is an inducible operon. It's something that can be turned on when needed. So this ARAC gene is kind of like an activator sequence. So this controls whether RFP gets made. The big idea is that transformed cells will produce RFP if there is arabinose. So if there's arabinose present, then RFP will get made, and therefore you will get these pink colonies, pink cells. Here is an animation of how this ARAC gene works. So ARAC codes for ARAC protein. So the gene codes for the protein. Arabinose is shown in the diagram. This is a sugar. So if arabinose is present in the medium, what will happen is arabinose will bind to the ARAC protein. If you guys noticed, that protein, it looked like a chunk disappeared, but it's a conformational change. It changed shape. So arabinose bound to the protein, now it's called an arabinose ARAC protein complex because they're combined together. This protein complex prevents DNA from looping, which allows RNA polymerase. It will bind to the complex and then helps it align to the promoter site, which is PBAD. Moving on, RNA polymerase then will transcribe the RFP gene to make the mRNA. mRNA codes for protein. Oops. And then the RFP protein would get made. This is actually what it would look like. So the ARAC gene, once again, codes for this protein, which acts like an activator, but only if arabinose is present, only if the sugar is present. Okay, so we have this recombinant plasmid, and then we have a culture of competent cells. So remember, I kind of mentioned earlier really quickly, competent means these cells have been treated so that it's more likely that they'll take in plasmids. So what would happen is you add the plasmid to these cells, and we zoom in, some of the cells will take in the plasmid. And this is called transformation. So transformation of E. coli with PARAR, that's the whole point of this experiment, is we're trying to get these plasmids in. Once again, it's kind of rare. It's not super common that this happens. Once the plasmid gets into the bacterial cell, now, in theory, it should be able to produce RFP and create pink colonies. So in the last part of the lab, this is the part where we're like trying to actually grow the bacteria. So what we would do is, it says growth and selection of transformed bacteria. So remember, not all bacteria cells will get transformed. So we only want to grow the ones that actually took the plasmid in. We have different plates. So these plates are agar plates. So if you looked at it, it looks like clear jello, but then it has added things to it. So this top row we call these P plus plates. The P stands for plasmid. So on these top plates, what we're gonna grow is we're gonna add in the E. coli that got mixed with PARAR. So really, if we go back to this animation, we're trying to grow all the bacteria that got mixed with plasmids. Some of them, go back into this, some of them will have the plasmid, some of them won't. So if you look at all of these cells, not all of them are gonna take the plasmid, only one of them will actually get the plasmid. So these P plus plates, we're gonna plate all the cells though that got mixed with plasmid. The P minus plates, so plasmid minus, is just E. coli. So these E. coli cells do not get mixed with plasmid. The different types of plates that we have, the first one is called LB. So on the bottom, I put a little key. So LB stands for Luria broth. Basically, it's just a broth of sugars and amino acids. It's nutrients for the bacteria. So it's like ideal growing conditions for bacteria. If I took an LB plate and just put it out in the open, after a couple of days, stuff will start growing all over it. The second type of plate that we have is LB/AMP. And AMP stands for ampicillin, which is the antibiotic. All right, so these plates, in theory, if I opened up this plate and just had it open in the environment, not as much stuff would grow on it because it contains ampicillin, which is an antibiotic. And then lastly, the third plate is LB/AMP/ARA, and the ARA stands for arabinose, which is the sugar. So if you recall, the sugar is needed to turn on the transcription for RFP. So what I would like you to do now is I want you to predict the growth of bacteria on each one of these plates. So you have 3 on top with the plasmid, and then you have 2 on the bottom that are not exposed to plasmid. Here are your options, your prediction options. So one thing that can happen is you have very high growth, and we call this a lawn. So if you look at this plate, this is just thousands of bacteria cells just growing. So they call it a lawn because thinking about the lawn in the front yard, it's just like a big layer of bacteria. We would say this is high growth. The second option is colonies. So this we would say is low growth because out of all of the cells that were in the solution, only a couple become transformed or will grow. So these are single cells that are able to survive. Now the low growth or colonies, they could either be white, really clear cells, no color, or they could be pink. So I want you to think about when you do your predictions, do you think if there is low growth, would they be white or would they be pink? And remember, they would be pink if they are producing RFP. The last prediction could be just no growth at all. So this plate, there's no bacteria growing, none of it grows. All right, so going back to this, I'm gonna go back to this one is, and you can pause and go back and forth, but I want you to sketch this out on your paper, and I want you to predict what you think would, how the bacteria will grow, either high, low, or none at all. And then whether or not, if there is low growth, whether there'll be any colonies. So let me start you off. Here are the options. Okay, this plate right here, LB, remember this is just nutrients. Okay, so if the E. coli plus the plasmid is grown on the LB plate, what you're gonna get is you're gonna get high growth. You're gonna get a lawn because the bacteria, they're happy. This is like nutrients, it's food for them, they could just grow. So this is gonna be my starting point for you, and I want you to try to figure out what you think will happen on the other plates. This one right here, these are also E. coli on Luria broth, so nutrients. So what do you think how do you think will grow? How do you think E. coli only will grow on a plate that has ampicillin, which is an antibiotic? So remember, antibiotics kill bacteria. How would you predict the growth would look like on this plate when it's incubated with the plasmids? And finally, on the final plate with the sugar too, arabinose. All right, so I will post the answer also later on, or maybe I'll just give you the answer when we do a class meeting, okay? All right, hope this helps.